Functional assays will help you reveal all the characteristics in your single-cell analysis. Hi, I'm Dr. Nora Fekete-Drimusz, and I work here at ARRALYZE, leading the bio team. Today I will talk about functional assays on the single-cell level. The FDA has recently approved immunotherapy against cancer based on NK cells, and this shed light on how heterogeneous these NK cell populations are. It also led to the question, how can we study these heterogeneous cell populations? How can we find the best cells in such a pool of cells? These individual cell characteristics are very important for future analysis. There has been a lot of improvement, of course, in high-throughput bulk cell analysis. This is definitely a step into the right direction, but we are still missing to be able to analyze large cell populations on the single-cell level. What would be needed is conditions close to in vivo and with as little stress on the cells as possible. The ARRALYZE platform with CellShepherd is aiming to provide a solution exactly for this question. With our machine, which we could actually call a cell laboratory on a footprint of 1 square meter, you are able to do 3 crucial functionalities. The first one is dispensing the single-cell solution very directly into these nanovalves. We at ARRALYZE are aiming to offer a solution exactly for this issue. And the answer for the question is the CellShepherd. With this machine, you can carry out all the 3 vital functionalities on a footprint of just 1 square meter. So first of all, the CellShepherd will let you dispense a single-cell solution into certain nanovalves. The glass nanovalves are inert, sterile, and they offer the best optical solution for microscopy. In the second step, you can image the valves individually. We offer 3 fluorescent channels and also bright field. It's also possible to do time-lapse imaging of your analysis. In the final step, you can extract valves of interest in a very gentle way, making sure that the cells remain alive for any downstream analysis. For NK killing assays, we use the CellShepherd to carry out a controlled co-culturing experiment. In this experiment, we had primary PBMCs, and we co-cultured them either with K562 target cells or Raji cells. To make sure that we can differentiate between these populations, the PBMCs were stained blue, while the target cells were stained red. Right after dispensing, they were incubated at 37 degrees with 5% CO2 and the right humidity. And we also added a cell death dye, cytox green, to the cell culture medium. The duration of the assay was 20 hours, and we took images continuously during this time period. What this did is allowed us to have a time-lapse video of these images. And the machine learning algorithm could then identify first the different cell populations, and then could also identify killing events that were marked by a color shift from red to green. This also meant that there were no false positive killing events recorded if a PBMC died. A really interesting thing that we saw during these experiments is how target cells don't die the same way. As you see in this video, even neighboring cells will be affected differently by the immune cells. Some will show apoptotic, while others show necrotic cell death. To be able to discover such a detail, you really need to have an immobilized single-cell solution. The only way you can shed light on such detail is if you focus on a very certain part of cell populations. And this is exactly what the CellShepherd allows you to do, with the nanovalves being there in the middle, immobilizing your cell suspension. The machine learning algorithm doesn't just record the killing events and their time, so giving you a kinetics of the killing itself, but it can also track the individual immune cells and show you how mobile they are, what their motility is, how much distance they have traveled during the whole analysis. All in all, we are able to provide an analysis of a killing assay that is much deeper than anything you have been able to achieve before. You get the kinetics of the killing events, the killing methods, and also gain some information about the individual mobility of the immune cells. All in all, we are able to provide you with a detailed analysis of your NK killing assay. You find out the kinetics of the killing events, the methods of the killings, and also the motility of each NK cell. But NK killing assays are not the only ones that could benefit from functional assays on the single cell level. In fact, anytime when you have a heterogeneous population, either coming from primary sources or after a cloning step, you need to look at how these cells compare to each other on the functional level. This is exactly what the CellShepherd would enable you to do. As long as your readout is optically visible in either of the 3 fluorescent channels or in bright field, we can provide you with the kinetics of your assay. If you also have such an assay in mind, I suggest you contact us and we try to find out together how to proceed.